Hello, my name is Justin, and today we're going to explore osmosis using something fun gummy bears. We'll see how water moves into and out of cells and how this affects living organisms. What do you think happens when gummy bears are soaked in water? What about sugar water? What about salt water? Take a guess. Do they get bigger, smaller, or stay the same? Hold your prediction or write it down, and we'll test it out later. First, let's talk about diffusion and osmosis for a bit. Diffusion is the movement of molecules from a high concentration to a low concentration, and the molecules get spread out. Osmosis is just the diffusion of water, but across a semi-permeable membrane and like the ones in real cells. Um, for example, water would move from a dilute liquid to a more concentrated solution with more solute than liquid. This is an example of diffusion until it reaches equilibrium or homeostasis. Um, Before we go into the key terms in the experiment, we're going to go a little bit more, just like a quick, brief overview about these biological examples. So these are some real biological examples that would be using osmosis. We have red blood cells in our body. When it's in pure water, it will burst because it swells. We have cells in salty water that will shrink because of more solute outside than inside. So it's like a hypertonic solution, which we'll find out later what it means. We have plant cells wilting in the hypertonic solution, similar to the solution situation of cells. And we have IV fluids. In hospitals, IV fluids always need to be isotonic to keep a balance, because if you do give the patient a wrong IV fluid, cells can swell or shrink dangerously. Point is, osmosis is important. So first, let's talk about some key terms. We have a solute. That solute is a substance that gets dissolved in a solution, which would be like salt. Uh, an example would be like salt, sugar. Solvent is the liquid that dissolves the solute and will surround the gummy bear, which an example would be like water, salt water, sugar water. The concentration gradient is a difference in how much solute is inside the gummy bear, in our case, versus outside the cup. If the solution outside has more solute than inside the gummy bear is in a hypertonic solution, which is basically more solvent than solute. In a hypertonic solution, water moves out. If the solution outside has less solute, the solution is hypotonic, more solute than solvent. In a hypotonic solution, water moves in instead. We can see an example of hypotonic and hypertonic on the top right. We can see the cell swelling from hypotonic and shrinking from hypertonic. Next, we have the selectively permeable membrane or the semi-permeable membrane, um, which is a membrane that only allows certain molecules to pass through but not others. For example, cell membranes let water pass through easily, but not large sugars or proteins. In our gummy bear experiment, the gummy bear isn't a real cell, but its gelatin behaves like a selectively permeable membrane. It will let water pass through easily, but not big molecules like the salt and sugar. Homeostasis, or is going to be seen in an isotonic solution on the top right, where you see water movement and the cell staying the same. And it's the process by which a cell keeps its internal environment balanced. Water moves in or out through the membrane to keep the inside just right, not too swollen, not too shriveled. These basic terms help us describe how water moves. Gummy bear actually works as a good model because its gelatin coating behaves like that membrane. So I kind of went ahead, but the next slide shows a visual of a gummy bear, and basically this is a gummy bear body, and it's basically gelatin, which will act like that membrane. The inside is mostly sugar, so water will move in or out depending on the environment. Different solutions will make cells shrink, swell, or stay the same, which is exactly what our gummy bears will show. If the environment has more sugar or more of the solute, water will move out. If the environment has more water, then water will come in. And then we have the isotonic solution. So this is the materials we need for our experiment. We need 3 gummy bears of the same color and brand. I'm gonna skip to the bottom um, to talk about control variables for a bit. So control variables are basically, or a constant is basically something that you keep the same throughout the experiment. So the reason why we want the same color and the same brand of the gummy bear, because even though it might not cause such a big difference, changing the color will add a new variable to the experiment, and you don't want that. You need to be sure that any change in the gummy bear size is due to just the water you are putting them in and not the color. So other control variables will be the temperature, the same soaking time, the same volume, and the same measuring method. So the 3 cups, we need 3 cups. So one for the water, one for the salt water, and one for the sugar water. We need a scale to measure the weight, and then we need a graduated cylinder or a measuring cup or something where you can measure the amount of water you're putting in. And then we need paper towels so we can dry the gummy bears before measuring. So the mass we record doesn't include the water outside the gummy bear, and so we could clean up. So for the procedure, for step one, we would label the cups, salt water, water, and sugar water. Step two, we would record initial weight of the gummy bears. Step three, we would use the graduated cylinder or measuring cup to get the same volume of water, salt water, and sugar water. And step four, we would submerge the gummy bears. Step five, we're going to wait at least 30 minutes or overnight for maximum results. If you have all the time in the world, overnight would be the best. Number six, we would remeasure and record observations and results. And number seven, we would compare changes. Uh, this is just a nice and simple experiment you can do at home with a friend or by yourself. Um, we would measure before and after. Remember to do that because you need to see the difference in size and weight of the gummy bears. So let's have some, before you start the experiment, I want you to write down and predict what will happen to the gummy bear in the following solutions: in the water cup, the salt water, and the sugar water. So you can brainstorm or discuss with a partner and write your guesses somewhere. What would happen to the gummy bear if it would like swell, shrink, stay the same, and maybe put why. And just do that first, and then we'll move on. So after discussing, um, remember to keep your predictions in mind and also that answer to the hook question. Um, so please go conduct your experiment, and then you can resume after you finish on the next day or in the next 30 for 1 hour, 30 minutes to an hour. And again, remember to record your results before and after the experiment. Good luck. All right, now that you are back, we can move on to the results. So after your experiment, your results should look like this. The gummy bear and the salt water should have shrunk or become lighter because it was in a hypertonic solution, so water will move out. The gummy bear and the sugar water would stay around the same weight or have some minimal change because it would be an isotonic solution, so the water is balanced. And the gummy bear in pure water should have been swollen and been heavier because it's in a hypotonic solution where there's more water outside and the water moves in. So on the left, we have an example, and we can see that the gummy bear over time in the hypotonic solution went from 1.70 grams to 8.09 grams. That's a difference of 6.39 grams. Um, yours might not be as drastic, but it definitely would have increased a good number or some number. So why does this happen? In pure water, water moves into the gummy bear. In salt water, water will definitely move out. And sugar water would match the concentration because the inside of the gummy bear is also sugar, so nothing changes. So let's piggyback off that uh, biological example slide and continue into the real world connection. So we can see three different examples. First, I'm actually going to talk about the IV fluids. I said at hospitals, IV fluids must be isotonic to your blood, which is again the same solid concentration inside and outside cells, and this would prevent cells from swelling or shrinking. Healthcare relies on controlling osmosis to protect cells, so it's very important in health. Speaking of health, we have dehydration. So dehydration disrupts osmotic balance in the body. When water levels drop, the blood becomes hypertonic, pulling water out of the cells. This prevents the body from maintaining the equilibrium that osmosis normally provides. As Lopez and Hall states, osmosis allows water and nutrient concentrations to be at equilibrium in all the body cells. So basically, this is an example of what dehydration does to our body and what we feel. So something other than health, we have food preservation. So I'm going to use pickling as an example, and the pickling involves the use of salt, which draws out moisture from the food through osmosis. So it involves dehydrating those cucumber cells with a concentrated brine and pulling water out. This would prevent bacteria and microbes from growing and preserves the pickle. And without osmosis, pickles wouldn't lose water and wouldn't last as long and spoil faster. So here are some reflection questions to think about. You can write the answers down on your paper below your results and just really think about it. So for one, how did gummy bears model real cells in this experiment? The second question, why did the gummy bears shrink in salt water? The third question, how would a red blood cell behave in each solution to kind of like connect the gummy bear to a red blood cell? And the fourth one, why is measuring mass a better indicator than length in this case? Because we could have also used a ruler if we wanted to measure everything, but why would mass be better than length? After reflecting, you have this, let's just have the main takeaway. Osmosis is water movement to balance concentrations. Remember that water naturally flows from areas of high water, of low or low solute to low water, which is high solute. Cells respond to their environment, just like gummy bears. Real cells will swell in water, shrink in salt, and stay the same in balanced solution. Osmosis basically keeps cells happy and balanced. These are my references, and thank you for following along. I hope you had fun doing the experiment, and if you want to do it again, you have more gummy bears. Enjoy, and thank you.